Welcome to BioBits and Bytes.
We have a really fascinating deep dive for you today. We're getting into the absolute essential infrastructure of life itself, the microtubule cytoskeleton.
It's so easy to think of cells as these little static blobs, but they're not. They're bustling, complex cities.
That's a perfect analogy. And if the cell is a city, then microtubules, we'll just call them MTs. They're the high-speed rail lines, the highways, and even the structural scaffolding holding up the skyscrapers.
But unlike our train tracks, these are constantly being built, torn down, and this is the really cool part, chemically tagged.
Yeah.
To direct specific traffic to specific places.
And that's our mission for this deep dive. We're going to unpack the interplay between the mechanics of these structures and their biochemistry, specifically how tiny chemical decorations, they're called post-translational modifications, or PTMs,
Right.
how they create what's called a tubulin code that basically dictates everything, the cell's shape, its stability, and where all the cargo goes.
I want to start with that structural part because these things are under a lot of mechanical stress.
Yeah.
So let's talk about the basic building block, the tubulin dimer. How does its shape matter so much?
Okay, so MTs are polymers. They're basically long, hollow tubes made of repeating units, and those units are the alpha-beta tubulin dimers.
Uh-huh.
But here's the thing. When a tubulin dimer is just floating around in the cell, especially when it's bound to GTP, it's not straight. It's it's curved.
So they're like little coiled springs just waiting for something to happen.
That's a great way to put it, a coiled spring. When these curved dimers get added to the microtubule lattice to make it grow, they are literally forced into a straight line.
And anytime you force something out of its preferred shape, you're storing energy. That's the conformational strain you're talking about, right?
Precisely. The entire MT lattice is just packed with the stored energy. It's not passive scaffolding at all. When the microtubule falls apart, that energy gets released and can actually perform mechanical work.
So the act of building the highway is also preloading a spring that can be used to push things around later. That is incredibly efficient. Have they actually measured this force?
They have, yeah. And it's amazing. A single growing MTN can push with about 2 piconewtons of force.
Which sounds tiny.
It sounds tiny, but you have thousands of these. It all adds up. And it's absolutely essential for cellular morphogenesis, basically, how cells build themselves into all the complex shapes they need to be.
Okay, so if the growth is so important, let's look right at the growing end, the so-called microtubule cap. The textbooks always show this perfect tapered funnel.
Right, that neat little schematic. And that schematic is probably one of the biggest points of debate in the whole field.
Really?
Oh, yeah. The idea of the cap is sound. It's a region rich in GTP tubulin that stabilizes the end. But when you actually look at it with something like cryo-electron microscopy,
Helium, yeah.
it gets messy. It's not a clean funnel. You see everything from just blunt, flat ends to these long, curved sheets that look like they're peeling away.
So there's no real consensus on what it actually looks like.
Not at all. It tells us that reality is just far more complex and probably asymmetrical than our simple drawings.
So what does that messy end tell us about how it all falls apart, the catastrophe?
It really hammers home the mechanical nature of it all. What we've seen from computer simulations and more cryo-EM is that the weakest links break first. And those are the lateral bonds, the ones holding the individual strands together side by side.
So the two unzips first.
Exactly.
Yeah.
The strands, the protofilaments, they splay outwards and curl up, releasing all that stored spring energy. And only then do the bonds along the length of the strand break apart to release the dimers.
Okay, hold on. Let's really unpack that. The whole system is designed to fail at its weakest points, turning that stored energy into the kinetic energy of these curling filaments. So you have these stressed, constantly changing tracks. But if they're all made of the same stuff, how does the cell decide which track is for what job?
And that brings us right back to the tubulin code. The cell overlays chemistry on top of this mechanical skeleton. It adds these little chemical flags, the PTMs, after the tubulin protein is made.
And these flags are like road signs for the molecular motors.
Perfect analogy. They're navigational cues. Let's start with one called detyrosination. It happens on the alpha-tubulin subunit.
Okay.
So tyrosinated tubulin is what you find in dynamic, fast-changing MTs. An enzyme comes along, snips off the end amino acid, a tyrosine, and now you have detyrosinated tubulin. And that's a marker for a super stable, long-lived track.
And I'm guessing this isn't random. Like in a neuron, for example, you'd see a specific pattern.
A dramatic one. It's fundamental to how a neuron sets up its whole structure. The axon, the long-distance cable, is full of these super stable detyrosinated MTs.
The long-haul highway.
Right. But the very tip of the axon, the growth cone that's actively exploring, is packed with the dynamic tyrosinated kind. The code literally builds the architecture.
Which has to feed directly into which motors can use which tracks. How do they know?
It's a fantastic question. The thinking is it subtly changes the surface charge or the physical shape of the docking site. So kinesin 1, a major cargo motor, strongly prefers the detyrosinated tracks. That basically keeps it and its cargo inside the axon.
But other motors?
Other motors, like kinesin 3 or kinesin 5, seem to prefer the tyrosinated tracks, which lets them travel in both axons and dendrites. The PTM is basically a gatekeeper.
That's amazing. Okay, what about the next PTM, acetylation? This one's location seems odd. It's on the inside of the hollow tube.
It is odd, isn't it? It's one of those beautiful little details. Acetylation is found on the most stable MTs imaginable, the ones in cilia and centrioles that have to last a lifetime. But since it's on the inside, in the lumen, it can't be a signpost for a motor walking on the outside.
So how does it provide stability? It's not a road sign. It's more like rebar inside of concrete.
That is a perfect analogy. Internal reinforcement. It doesn't seem to make the MT more or less likely to grow or shrink. Instead, it seems to protect it from mechanical aging.
Wear and tear.
Exactly. All the bending, the stress, the friction of motors running over it. Acetylation is thought to make the structure more flexible. It reduces friction between the protofilaments and helps the microtubule self-repair. It just makes the tracks last longer.
So it's a mechanical safeguard.
Yeah.
But you see these contradictory studies, right? Have you ever wondered why, in a test tube, acetylation doesn't seem to affect kinesin 1's speed, but in a living cell, it seems to help it run for longer?
That's a great question. Let's break it down, because that contradiction tells you everything about the difference between a simplified system and a real messy cell.
Okay.
In the test tube, the motor is just running on a single track. It can't see the internal modification. But in the cell, the protective effect of acetylation helps microtubules form bundles.
Ah, so it's not a better track. It's a multi-lane superhighway.
You got it. The motor doesn't run faster, it just runs longer because it's on a much more robust bundled highway and it's less likely to fall off.
That makes so much more sense.
Oh.
Okay, let's get to the last big one. Polyglutamylation, the external traffic controller.
This one is absolutely critical, especially in neurons. So this PTM involves adding chains of another amino acid, glutamate, onto the C-terminal tails of tubulin.
And these tails, where are they located?
Crucially, they stick out from the outer surface of the microtubule. They're right there, accessible to every motor protein, every binding protein, every enzyme that interacts with the track.
So you have enzymes that add the chains, the TTLLs, and enzymes that remove them.
Right, the CCPs. And there's even specificity there. In neurons, TTLL1 tends to polyglutamylate alpha-tubulin, while TTLL7 does beta-tubulin. And this really shows you how the code gets read. Remember, acetylation, the internal one, motors pretty much ignore it. But polyglutamylation, this external one, it regulates almost everything that touches the MT.
So how does that regulation actually work? What does it do to traffic?
Well, in lab experiments, it's pretty clear. More polyglutamylation makes the protein tau stick on much harder. It also revs up the activity of catanin, which is an enzyme that literally severs microtubules.
That chops the tracks up.
It chops the tracks up. And for motors, it's even more specific. Kinesin 1 motility seems to be affected only by the polyglutamylation on beta-tubulin, not the stuff on alpha-tubulin.
But the truly critical experiment is seeing what happens in a real neuron when this system breaks down, when you get hyperglutamylation.
This is the experiment that changed the field. When you delete the enzyme CCT1, the one that removes the glutamates, you get way too much polyglutamylation. And what they saw was a generalized system-wide reduction in cargo movement.
Let's pause and simplify that part again. We're talking about everything. Mitochondria, lysosomes, vesicles, things driven by totally different motor families like kinesin 1 and kinesin 3.
Everything. Everything slows down. They spend less time moving.
Wow.
And that universal slowdown, that tells us that polyglutamylation isn't just a signpost for one motor. It's a broad master traffic control mechanism. And when you mess it up, the whole highway system grinds to a halt.
And this isn't just some abstract finding. This has devastating real-world consequences.
It absolutely does. The loss of that CCP1 enzyme, which causes this hyperglutamylation, is directly linked to a severe infantile-onset neurodegenerative disease. This traffic jam is now seen as a central cause of the disease.
So zooming out a bit, MTs aren't working in a vacuum. They have to coordinate with the other major part of the cytoskeleton, the actin network.
Right. The two systems work together perfectly. MTs are the long-haul, high-speed fracs. Actin filaments are the local roads, a dense meshwork right into the cell surface for short-range delivery.
The high-speed train and the final mile delivery van.
That's the perfect way to think about it, and you see it most clearly in places like the neuronal growth cone.
Yeah.
The actin network at the edge builds the scaffolding that the MTs then grow into, guiding them to their target. And they're all linked together by a whole class of proteins called MAPs.
Microtubule-associated proteins. And they add yet another layer of control, right? They can act like speed bumps or accelerators.
A crucial layer. Some MAPs are just roadblocks. Tau, which we know is a big player in Alzheimer's, can physically get in the way of motors. But other MAPs, like ensconsin, do the opposite. They actively recruit kinesin 1 to the track, basically making sure the engine is ready and waiting for cargo.
Let's look at a really vivid example of this kind of macro regulation, the color-changing cells in fish and amphibians, the melanosomes.
Oh, it's a beautiful system. The movement of the pigment granules is controlled by hormones that change the levels of a simple chemical signal inside the cell, CMP.
And how does KNP control whether the fish is light or dark?
It's all about which motor is active. When a hormone like melatonin drops KNP levels, it activates dynein motors, which pull all the pigment to the cell center. The animal looks dark.
And the opposite.
The opposite happens when a different hormone raises KNT. That activates kinesin 2 motors, which spread the pigment out all over the cell. And the animal looks light. The whole cell just flips a switch.
But even there, the MT highway needs the local actin roads for the final stop.
Absolutely. Once the pigment is dispersed by the MTs, a complex of proteins has to tether those pigment granules to the actin network at the very edge of the cell. If you disrupt that tether, the pigment can't stay put, and you get a lightning effect. The cargo gets delivered to the right neighborhood, but can't be dropped off at the front door.
So let's circle back to that huge question about the neuronal traffic jam, the one caused by hyperglutamylation. What's the leading hypothesis for the mechanism? What's actually causing that universal slowdown in the cell?
That's still the big unknown, but there are two really strong hypotheses, and they both connect to what we know from the simpler experiments.
Okay.
Hypothesis 1 is the sticky roadblock idea. The excessive polyglutamylation makes inhibitory proteins, especially tau, bind way too tightly to the microtubules. The tracks get covered in these sticky tau proteins, and they act as roadblocks for every motor, slowing everything down.
So the road is fine, it's just covered in traffic cones. What's the second idea?
The second is the fragmented track hypothesis. This idea is that all that polyglutamylation sends the severing enzymes, like catanin, into overdrive.
Yeah.
So they just start chopping the long, continuous highways into tons of tiny little segments. The motors can't get a good long run. They have to constantly detach and reattach, which would kill their overall progress, even if their speed on the little fragments is normal.
This whole deep dive just shows how unbelievably dynamic and, well, intelligent these cellular structures are. If we were to summarize the key takeaways.
I think number one is that microtubules are fundamentally mechanical stressed polymers. They store energy.
Right, and their function is totally governed by this chemical code of PTMs. So detyrosination sets up the stable highways and axons.
Acetylation is the internal reinforcement, the protection against wear and tear.
And polyglutamylation is the master traffic regulator. And when it goes wrong, especially in neurons, you get a system-wide traffic jam. And here's a final thought for you to take away. These microtubules are not passive scaffolds. They're actively managed. They can self-repair. And what's more, in response to major cellular stress, the cell will deliberately hyperacetylate its tubulin to trigger autophagy, the cell's own cleanup and recycling system, as a survival mechanism. This intimate link between the highway system's integrity and the cell's survival makes the cytoskeleton one of the most critical and I think most exciting therapeutic targets out there.
Thank you for joining again.